Hey guys, I'm going to be showing you a video about how DNA is used to make RNA, and then how RNA is used to make protein, and then we'll go into some genetics vocabulary. All right, the first thing is the structure of DNA versus RNA. This is a little bit of a review, but over here on the left we have DNA, deoxyribonucleic acid, and you can see here that it has two strands, whereas on the right, RNA or ribonucleic acid has only one strand. The other difference here is that in DNA, it has thymine as a nucleotide, whereas RNA has uracil. All the cytosine, adenine, and guanine are the same for both RNA and DNA. And here we have a restatement of that. So DNA contains the genetic information for almost all organisms and contains segments known as genes, and they carry the code for various proteins. And the base pairs are A, T, G, and C. A pairs with T, and G pairs with C. And for RNA, RNA is ribonucleic acid, and that holds a code for a specific protein to be made, and the nucleotides there are A, U, and then G, C. A and U correspond with each other, and G, C. The process of DNA replication is when DNA copies itself to make two identical copies, and this is done before a cell can go through asexual reproduction. And we'll talk about asexual reproduction in just a bit. And this takes place in the nucleus of the cell. The DNA is in the nucleus, and DNA replication takes place in the same place. DNA replication is pretty straightforward. What happens here is the parent DNA on the right is unwound by DNA helicase, and then the strands of DNA are copied, and so what we have at the end are two strands of DNA that are identical to the original strand. All right, so all of that so far has been review. Now we have new stuff. RNA transcription is the process by which a segment of RNA is made by reading or transcribing a gene or a segment of DNA. And this also takes place in the nucleus of the cell because it uses DNA, and DNA is found in the nucleus. So what happens here is an enzyme called RNA polymerase travels along the DNA and unwinds the DNA, allowing for a strand of RNA here to be used or to be transcribed or made by reading the strand of DNA. The next step is translation. So what happens after the segment of RNA is made, it leaves the nucleus and heads out to the cytoplasm, and it either takes place in the cytoplasm at a ribosome or at the rough endoplasmic reticulum, also using a ribosome. Okay, so what happens in translation is that a ribosome links amino acids together to form a protein. All right, so here we have an image of a ribosome translating a protein. So here we have the mRNA strand, and you have C, G, U, G, A, C, A, G, for example. These are all of these nucleotides are in here. And you'll notice here that there's a 3-letter segment here, A, C, G. And what that does is it brings in this molecule right here. This is called a tRNA, or transfer RNA molecule. This has a 3-letter code that corresponds to this 3-letter code. The one on the RNA is called a codon. The one on the tRNA, on the transferred RNA, is called an anticodon. And what it does is it brings in these amino acids, and it then, as a chain forms, that makes a protein. ACG, CGU, and AGU. Once again, the codons are 3-letter nucleotide segments on the RNA strand, and these are used to make to bring in specific amino acids to make a growing protein. Okay, so notice on the previous slide the codons were ACG, CGU, and AGU. Okay. ACG, if we were to use these charts, we could figure out what amino acid that is for. ACG, right here. We look at the first position, A, then we look at the second position, C, and then we look at the third position, G. And so ACG is right here, and that'd be threonine. CGU, using a different color, CGU would be arginine. And AGU, using a third color again, AGU would be serine. And that's how it determines which amino acid to bring in, and you'll have to learn how to do this as well. So this amino acid would be threonine, this would be arginine, and this would be serine based on the codons that we see down at the bottom. And so this growing protein over here on the right, okay, the amino acids that are brought in, we find the code for those down here on the RNA. So again, just an overview of the protein synthesis. In the nucleus of the cell, we have DNA. That DNA is being unwound and a segment of mRNA is made. Then that mRNA leaves the nucleus and heads out to a ribosome. At a ribosome, tRNA molecules are brought in and a chain of amino acids is formed to form a protein. So DNA is transcribed to make RNA, and then the mRNA is translated to form a protein. So what do these proteins do? So the genes code for different proteins to be made. So what? Why should we care? Every single one of your physical traits that makes you unique is caused by different proteins that are made by reading your DNA. Your brown eyes are caused by proteins. Your black hair, caused by proteins. The ability to roll your tongue or have a hitchhiker's thumb or a widow's peak, detached earlobes, all of it is caused by proteins.